Hello everyone. In this session we are going to discuss about interaction of genes, which comes under the paper 6, that is genetics, biostatistics, evolution and paleontology. So under this topic already we have covered the different types of interaction which comes under the non-allelic gene interaction. So today in this session mainly we will be concentrating on allelic gene interaction. So here the examples are complete dominance, incomplete dominance, codominance, pleiotropism, penetrance, expressivity are the again multiple genes. So these are the some of the examples which comes under the allelic genic interaction. So coming to the first example under allelic interaction, that is complete dominance. So complete dominance is nothing but the two genes which controls a character. So those what happens, they are called as allele. So in that two alleles, one allele expresses its character in the first filial generation or the F1 generation. So that is called as the dominant allele. So here that dominant allele will be expressed in the first generation. So this ratio what we call that comes under 3:1, because 3 will be dominant and 1 will be recessive. So in where it happens, it takes place in the monohybrid cross. So already we have discussed about the monohybrid cross by the Mendel. So first example we had taken, tall plant is crossed with the recessive, but here I have taken the example of purple color is crossed with the white flower. So here what happens, all the 4 individuals in the F1 generation will be purple color because of the dominant allele which expresses its character as a dominance. So that is how in the first filial generation we will get all the flowers or all the traits, that is the dominant. So that is about the complete dominance. So example, you can tell F1 tall plant in the Mendel experiment or the coloration in pea plant as given as the example. So next example is incomplete dominance. So here what happens, the F1 hybrid have an appearance somewhere in between the phenotypes of the two parental individual. Like for example, if you take the red and white color, so in between after mixing red and white color, a term like a pink coloration comes. So you can tell that is one more color. So here the same example they had given, color flower color of the 4 o'clock plant, that is Maribellis jalapa. So example, so here what happens, the incomplete dominance in a allelic interaction, what here the both allelic that is a character expresses their character in the F1 generation. So both alleles, it may be a dominant or the recessive. Then we'll get the example, that is here, that is in incomplete dominance what happens, if you are taking capital R and capital R as a flower color, that is dominant flower color, which will be red or the purple in color. Again small r which I am taking as the white in color, that is recessive. So once there is mixture of the capital R with the small r, then we'll get the F1 hybrid as capital R and small r. But here what happens, there will be a mixture of the capital R and small r. So due to that we will be getting the terms, that is the first one as capital R and capital R, second it will be a mixture capital R and small r, later we will be having capital R and small r, and at last we will be having small r and small r. So here what happens, there will be combination of dominant as well as recessive. That is how the combination of both the coloration takes place, and that leads to the formation of pink color, or you can see the lavender when it is coming to the purple. So this is how incomplete dominance takes place. So this is the complete theory related to the Maribellis jalapa, how it takes place in or the 4 o'clock plant. So next, so this is how the coloration takes place in the nature when you just mix the red color and the white color, which leads to the pink color. Next we are having codominance. So codominance is nothing but here the both alleles of the character are equally dominant. So incomplete dominance was nothing but both will be expressing, but here codominance. So here also somewhat the terminology will be same, mixturing of between dominant and recessive, but codominant means one can be a more dominant and other can be the one more, but here none of them is masked, means one will be more dominant, one will be recessive. That can't happen. But here what happens? Both will be dominant acting. So that is how we call it as codominance. So example, that is codominance is a allelic interaction. Here what happens? The in heterozygote, both allele expresses their character simultaneously. These genes are called as codominant gene. So example is the color coat in shorthorn cattle, ABO blood groups, MN blood groups, sickle cell anemia. So these 4 examples I have noted in this, but usually they will ask 2 examples for the examination point of view. So color coat in the shorthorn cattle. So as I have taken the capital R and capital R, they are the red colored, the shorthorn, and small r and small r, I can take it as smaller, or the capital dash and r dash and r dash, that I have taken the white. So when they both are combined, here I have taken, okay, it is smaller, but the example in this I have denoted as the r dash and r dash. So when both capital R and small r are combined, the F1 individual will be capital R and small r. Later in the both, or if you want to check for F2 hybrid, so there what happens, the combination of capital R and small r, it takes place, and that will lead to the formation of the roan color. Roan is nothing but it is a mixture of the red color and the white color. So usually roan, as you can see in this picture, the red colored shorthorn cattle, which will be having the white colored patches on its body. So this is how the first example, inheritance in coat color of shorthorn, that is in cattle. So second, ABO blood group. So this blood grouping we will be studying in multiple allelic interaction also in detail. So here I have mentioned just how the blood groups are that act as codominant. So we know in humans we are having 4 different blood groups, that is A, B, AB, O. So here the homozygous A group has the genotype as LA and LA. And later the gene LA produces the antigen A in A group. So usually LA produces antigen A, that leads to the formation of blood group A. Similarly for B. So here homozygous B, that denotes, or it will be seen in LB, which produces the antigen B, then that will be B blood group. So now a scenario or the example, when A blood group is married to B blood group. So what happens? So here the A blood group man is married to B blood group woman. So there the combination of both antigen or the genes LA and LB takes place. So that leads to the formation of the AB blood group. So AB blood group will be formed. So later in F2 generation, the F1 is crossed with F1. So we know that F1 we are having already LA and LB. So now when LA and LB are crossed with LA and LB, so what happens? LA which will give you the A blood group, LALB will be having B blood group, LA and LB again B blood group, and later we will be having LB and LB, which leads to the formation of B blood group. So this is how the blood groups takes place in the ratio of 1:2:1. So 1 will be A, 2 is AB, and B will be 1. So O blood group, how it takes place? We'll see the example in this picture. Like, see, the antigen A, if it is present, and anti-B antibody will be present in the plasma, that leads to the type, that is blood group A. So usually they cannot have B or AB blood group, but they can have A or O. So before that we need to see in antigen B what happens. Anti-B antibody will be present, so that is why they cannot have A now or AB, but they can have B or O blood group. So during donation of the blood group, so what type of antigen and what type of blood group they will check before, then only the blood donation they will take place. So and antigen A and B, they don't have anything, so they can have any type of blood group. So AB is very good, and later in O or neither A or B, so that will be O. So type O blood group, they can have only O group, so that is the universal donor. So they can donate to everyone, but they cannot take. So that is how usually O blood group, they are called as universal donor, and AB will be the universal, the recipient. So already we have saw this example. Now coming to Mn blood group. So what is this Mn blood group now? So usually this Mn blood group is classified into three types: M, N and Mn. So they are also notated by LM and N, LM in M, and N in that is LN and LN, and Mn will be LN and LN. LM and LN will be the Mn blood group. So here the blood group that takes place, that is M group, will be having LM and LM, and N group will be having LN and LN. So that leads to the formation of Mn, that is it will be having LM with LN. So later when LM and LN is crossed with each other, that is F1 is crossed with F1. So in F2 generation we will be having, that is first blood group will be M. So that contains the M group, and second we will be having M and N. So that is LM and LN. Later LM and LN, that is Mn blood group, and at last we are having N blood group, that is LN and N. So usually this is also a type of example that comes under the codominance. Coming to last example, that is sickle cell anemia. So already you have studied in your lower classes what is the sickle cell. So usually our RBCs are round in shape and they are somewhat bulged, but these sickle cell, these cells, they are affected by the genetic disorder, and the cells will become half moon shaped or the crescent moon shape. So that is how the sickle cell anemia is caused. So this is a type of hereditary blood disease. So there what happens, presence of sickle shaped RBC that takes place under the low oxygen pressure. So here in sickle cell anemia what happens, the first what we can see, the HbS and HbS, that is sickle cell anemic person, and later this HbS produces the defective hemoglobin, that is Hb. So later the normal person will be having Hba and Hba. So HbS will be sickle cell person and normal will be Hba. So now later if they marry to each other, then what happens, HbS will be combined with Hba, then they will become the carrier. So here the example tells that when the unaffected carrier father, so carrier is nothing but HBS with HBA. So he will be a carrier. So unaffected carrier mother. So even she is the same thing, HBS with HBA. So now when they combine HBS, that is crossed with HB, yes, so what happens? Both will be, they contain S now. So due to the presence of S, so that will be what? That will be affected to the main, this person. So usually that person will die. So this person will die. So and due to the presence of HBS and HBA now. So as I was telling, the scenario, usually both they will carry what? HBS with HBA. So when they are crossed with each other, that is HBS and HBA. So what are the chances there? HBS with HBS, HBS with HBA, HBA with HBS, HBA with HBA. So 4 it will takes place. So HBS with HBS will be this person who is the affected one. HBS with HBA, HBS with HBA. So that will be the unaffected, but they are carrier. HBA with HBA, so this denote will be the blue in color because he will be unaffected. So this is how the controlling of the sickle cell anemia or genetic type that takes place in the sickle cell anemia. So next, coming to pleiotrophism. So it is a very important type of genetic movement or the allelic interaction. Here what happens, usually the production of many character by a single pair of gene is called as the pleiotrophism. So it is a multiple effect. So what happens, see, as you can see, the beta globin gene mutation takes place that leads to the pleiotrophism, and that leads to blindness and liver failure and one more, that is heart attack. So one gene is changed but many disorders are coming. So that is called as the pleiotrophism. So later, so here what happens in pleiotrophism, the single pair of genes control many character, but in same, that is small p and small p in phenylketonuria produce the accumulation of phenylalanine in blood. So phenylketonuria is also a type of genetic disorder where what happens, the tyrosine will be produced. That time the hydrolyzation of the phenylketonuria will not takes place. So that leads to like mental retardation, widely spaced incisors, and the pigmented patches on the skin, excessive sweating, non-pigmented hairs. So these all takes place. That is how this phenylketonuria will be taking place. So next, coming to the penetrance. So penetrance, here what happens, the percentage of the individual expressing the character for a particular genotype, that is called as penetrance. So there are of two types of penetrance, that is complete penetrance and incomplete penetrance. So in complete penetrance what happens, the expression of a character for the particular gene that takes place, that is called as complete penetrance. But in incomplete what happens, only few individuals do not express, but whereas in complete all the individuals will be expressed. So example is blue eyes. So as you can see in the picture, like blue eyes, how they usually change in terms of the genotype. So genotype capital T and capital T, that will be the example for the Mendel's tall plant. But when coming to the capital B and capital B, they produce blue eyes. Here 90% of the human beings which are having capital B and capital B, that gene will be responsible for the production of blue eyes. But there is also a chance of only 10% of the people having white eyes, even though they contain the gene capital B and capital B. So here what happens? The expression of the blue eye takes place, and that leads to the expressivity. So this penetrance is influenced by the environmental factors such as food, light and temperature. So expressivity and penetrance, they are two different types of interaction, but they can be occurred within the same gene also. So here in the expressivity next, so the variation in the degree of expression of a particular gene, as given the example, that is the vestigial wing in Drosophila. So here this expressivity is also due to influence in environmental factors on genes. So here as you can see in the picture, they are of different types of Drosophilas, and they are having mainly concentrate on the wing pattern or the wing size. So in wing size, the first one, the wild type. So in wild type what happens? As you can see, the wing is much elongated and it is larger than the body size. But now just go and see the vestigial wing or the curled wing. So curled wing is nothing but same, the size will be the larger, but the wings are curled, but mainly concentrate on this vestigial wing. So here the wings are much reduced and they are very much smaller when compared to the wild type. So this is how the expressivity takes place on the different types of phenotypes of Drosophila. So here the recessive gene the recessive gene will be small letter. So recessive gene small v, where the vestigial wing will be developed in the recessive flies. So that is how small v has to be expressed. Then when you grow these flies, so they are grown in the normal room temperature. So that time the flies develop typical vestigial wing. So remember the temperature, that is 72 degree Fahrenheit. So now when they are grown in 80 degree Fahrenheit, the wings are slightly longer. So it means they will be normal type. And again these they are treated in the different room temperature like 88 degree Fahrenheit. So what happens? The wings are still longer, extending from tip to the abdomen. So they are the largest when they are kept in 88 degree Fahrenheit, but when they are kept in 72 degree Fahrenheit, the wings will be in the vestigial form. So this is how the penetrance and expressivity that takes place in genetics. And here I have said the percentage of individual having a particular genotype that expresses the phenotype. That is true, because the genotype is much very important for the penetrance and expressivity. So here in the expressivity, the degree of which the character is expressed. So when the character has to be expressed, then only the expressivity also will takes place. So this is about this session. So in this session we have discussed about complete dominance, incomplete dominance, then penetrance, expressivity. So very important for your examination. So here they can ask the examples like blood grouping type and short horned coloration. Then what do you mean by penetrance and expressivity with examples. So this type 2 to 3 mark question can be asked from this session. So that's all about this session. Thank you.